based in Ian's lab and Abby's lab, and I've been here for about a decade now. And for the last 10 years, I am doing research, but I'm also like really the lab manager and a system administrator. And one of the ways that I assess the quality of grad students in the lab is really how much grief they cause me. Because sometimes, I arrive work on Monday morning and some grad student has just decided to try and download GenBank and the disks are full and headaches and problems. So by that reckoning, Abby is a model student. Abby's never given me any grief over the time she's been here, which is fantastic. And also, I think, like many students in our lab, they arrive, and most grad students, when they come to grad school, are nervous presenters. And Abby is one of the many students in our lab who's really learned the art of presenting and telling a story. And at such a point, I think everybody in our lab would be like, Abby is speaking next week. You've got to go to Abby's talk. So Abby always gives these amazingly, no pressure. Abby gives these amazingly visual, rich talks, which we always look forward to. And Ian will say something now, but yeah, we're going to miss Abby in the lab, and good luck with everything, Abby.
Thank you.
So graduate school is generally a time of great change. Students not only learn about science, but about themselves. They learn how to organize, conduct, and present research. They teach as much as they're taught. Some graduate students anyway. There are others that find the experience quite miserable because the life of a scientist is actually not for everybody. I'd like to say that Abby blossomed under my care, but it's not really true. She didn't have a lot of growing to do. She was already in full bloom when she got here, and I'm proud to say that my lack of watering didn't kill her. I would say that she thrived despite my laissez-faire attitude, not because of it. I think she has learned a few coding tricks from me, and I can take credit for that, but not much else. She's always been very organized in her research and lots of attention to detail, the kinds of things that we like to teach. And as Keith was saying, we've really been in awe of her sort of aesthetic sense from day one to now. And that's one of the things we're going to miss about her is that, when everybody gives a talk and then she has some idea about how to make it better, it's always a great idea. I've really been honored to have her represent my lab because she makes us look better. And it's great that she's already got a job that she's going to. I'm very certain that her new employers are going to like her as much as we did and do, and I'm hoping she'll come back and visit us sometime. I think my only regret from the time that she's leaving now is that when we were both playing a lot of WoW, we never got in the same realm of PvP together. Damn. Anyhow, so let's turn it over to her research and see what she's up to.
Thank you. It's actually probably better that we did a PvP because I was always the guy on Rapid Base and I would just get knocked off the cliff and it'd be like, good luck, Ian, while you're just getting like jumped by 5 rogues that were hiding there.
Not everybody needs to know that.
You're losing the audience.
So I'm going to talk about something that you all know about, which is some of the science that I've been working on during my career here at UC Davis, and it's been a real fantastic journey, full of ups and downs, many more ups and downs. And really what I want to focus on today is a selection of the projects I've had the privilege of working on, specifically the bioinformatic methods we've developed for understanding engineered DNA binding protein specificity and their application in malaria research. In short, let's not advance the slide. Okay, yeah, woo, science. All right, let's get started. So just a really quick look on what's going on. I'd like to start off with a bit of a prologue, give you a brief history on genome engineering and where it was and how we got to where we are today, before jumping into chapter 1, which admittedly is a little bit more background as well, talking about the molecular tools we use for genome engineering today. Then I'll actually jump into one of the projects that I worked on, which was developing artificial transcription factors for malaria research. And then finally I'll wrap up with Mermaid, which is actually kind of going in reverse chronological order, but Mermaid was a software package that Ian and I worked on for de novo motif discovery for DNA binding proteins. So starting off with the prologue and the genome engineering lore, if you were to go to Google and Google genome engineering, it would tell you to go to Wikipedia, and then Wikipedia would tell you that genome engineering refers to the strategies and techniques developed in recent years for targeted specific modification of the genetic information, or the genome, of living organisms. And that's a pretty good definition of what genome engineering is. Back in the day, there were techniques that were available where you could insert genes of interest into another organism, and this was largely accomplished through, say, horizontal gene transfer. So for example, you might use an agrobacterium which contains a plasmid containing your gene of interest, or you could use a lentiviral vector. But essentially, both of these systems would transfect or transduce into your target cell, and then they would have non-specific integration, I'm going to use this laser pointer, non-specific integration into of your desired gene into the genome. So this wasn't quite meeting what the current definition of genome engineering was, because it's not specific. We know what gene we're putting in, but we don't really know where it's going. On the other side of the spectrum, there is random mutagenesis, which if, say, you didn't really want a gene that you specifically wanted to put in, but you just wanted to mutz around a little bit and see what happens. So you can start off with this helpless seed here, or an embryo, or whatever you want, and then you can irradiate it, or you can expose it to some chemical mutagen, like EMS, and then you get a variety of phenotypes. A lot of them will probably be stunted, assuming they lived at all. Other ones might look, well, this is supposed to be Arabidopsis, an abstract version of Arabidopsis. Some of them might look pretty close to the normal wild type. Other ones get a little weird looking, kind of some strange phenotypes start showing up. And occasionally you get one that's really interesting. And then for those super interesting ones, say like I wanted to pick this piranha plant, and you, being a geneticist, you want to figure out what genes did I change to get that phenotype? How did I get from an Arabidopsis to this thing that terrorizes Mario? And the fact of the matter is that you could probably find the change that caused that phenotype. But one of the side effects of random mutagenesis is that you caused a whole bunch of other double-stranded breaks in the genome that just kind of end up as collateral damage as part of the method. So when the cell is faced with so many double-stranded breaks, it really has one of two ways that it could repair them, because double-stranded breaks are a really catastrophic event as far as a cell's survival is concerned. The first method, non-homologous end joining, is really kind of cut and dry, and it's also very brute force. So you start off with your double-stranded break, or DSB, and it can result in either a deletion, where you just lose the genetic information near that double-stranded break, or it can also result in random insertions. Both ways do modify the way that the genome is ultimately read, because it either throws things out of frame or you lose information. A more sophisticated method that's available to people, not people necessarily, but organisms that have more than one copy of their genome. So people like organisms like us, we have two copies. Homology-directed repair is more sophisticated. So in the event of a double-stranded break, instead of just taping it back together, you could have a homologous template, which is basically a long stretch of DNA with sequence similarity to the damaged region. And then with some molecular gymnastics involving strand invasion, D-loop formation, and DNA synthesis, it eventually resolves itself where you've now replaced the broken piece with a copy of the homologous template. So you are able to better maintain genetic integrity with this pathway. So knowing this and knowing how the cell uses homology in order to repair its own double-stranded breaks, these handsome fellows in 2007 got the Nobel Peace Prize for using homology-directed repair to create specific modifications in mice. So Kopecky, Evans, and Smithies came up with a method that involved using a donor plasmid with homologous arms. So in the event of a double-stranded break, you also donate this plasmid that contains a sequence of your choosing, flanked by very long stretches of homology. And so with the presence of homology, the cell will then utilize that homology and then do the repair pathway, except in the process it will actually integrate your sequence into that targeted region, as defined by the homologous sequence. So this was a huge breakthrough. This was big because now you weren't relying on random integration events. You weren't relying on just getting lucky and getting it to where you wanted it to go. However, spontaneous integration events were extremely rare. Like I'm talking really, really rare, like super rare, about 1 in a million rare. And so this requires a lot of screening that really just wasn't feasible from the standpoint of wanting to go into high throughput. To find that single event, you had to dedicate so much time to just finding that needle in a haystack. So of course I'm sure someone out there had said, there's gotta be a better way. And sure enough, there was. So this idea of molecular scissors and how this advanced genome engineering as we knew it. A big question that people had was that if we could make a targeted double-stranded break, that might help things. That might facilitate and drive homology-directed repair. So is there a way that we can intentionally introduce a double-stranded break instead? And then we follow the same protocol where we give it the donor plasmid with a homology and it gets integrated in. And it turns out that when you do this, your chances actually shoot up to about 1 in 100 on a good day. So that's a huge improvement. That's like a, I'm not good at math, 10,000-fold increase, which is really, really amazing. And so now, instead of using like boxes and boxes of screening plates, you can just do everything on like a single 96-fold plate, and chances are you'll get the insertion that you want. So that was great. And then the question became, how do we do this? Or more importantly, how do we do this reliably? And the answer is that you use one of many families of engineered nucleases. The first and the oldest family would be the meganucleases, the zinc finger nucleases, the tail nucleases, and the CRISPR-Cas9 system. So just out of curiosity, how many of you guys have actually heard of CRISPR-Cas9? How many of you guys use it? Of course my lab does. All right, good job, guys. So, kind of a brief overview on what the pros and cons of these platforms are. Meganucleases were nature's molecular scissors. So they were these super specific endonucleases that just occurred in nature. So obviously scientists would think that's like, okay, well, you know, if nature wanted a site-specific nuclease, then these would probably be it. And they recognize a 14 to 40 base pair sequence, which is really long as far as a recognition site goes. However, they're very difficult to reprogram. There never really came a recognition code where people could predictably change or mutate the protein in a way that you could reprogram the endogenous target site. So because of that, meganucleases never really took off. Then came along zinc fingers, or zinc finger nucleases. So I should say that the zinc finger domain here, these 3 boxes here, is DNA binding only. However, you can attach another domain, say a nuclease, like a Folk1 nuclease domain, to make it the zinc finger nuclease. But essentially, these are chimeric proteins. And zinc fingers are nature's, not like meganucleases, they're also nature's favorite, except they're eukaryotic transcription factor motifs. So they're the most commonly occurring transcription factor motif you find in eukaryotic genomes. And unlike meganucleases, these actually recognize things in multiples of 3. So each finger recognizes 3 bases at a time. And you can rearrange them to change the targeting behavior of these zinc fingers. But the assembly can still be a little difficult. As your fingers get longer, the predictability of where they go starts going down. Transcription activator-like effectors were making a big splash when I came in to join Dave's lab in about 2010-ish. And because originally I come in and I said, like, I want to work on zinc fingers. And he's like, forget zinc fingers. There's this new thing called transcription activator-like effectors. You should work on that. And I'm like, okay, Dave, that sounds like a good idea. So I thought I was being on like the bleeding edge of genome engineering research. And so transcriptional activator-like effectors, or tails. I'm just calling them tails for now. In nature, they're this bacterial transcription factor for pathogenesis, where they're inserted into the plant host cell and they bind target promoters in order to change the cell's physiology to make it more hospitable to the bacteria. Like zinc fingers, they're actually DNA binding only, so again, this is a chimeric protein. And they recognize a pretty long recognition sequence, between 12 to 30 base pairs. I've heard some of them get a little bit longer. And they recognize, each domain recognizes it 1 at a time, as opposed to zinc fingers, which each finger would recognize 3 bases at a time. But more importantly, these are easier to reprogram than zinc fingers or meganucleases. So there's this explosion of tail assembly kits that you could use with overlapping ends that you can just, with still some slightly specialized molecular biology, you could assemble tails in a very systematic fashion. And then about a year and a half ago, CRISPR-Cas9 came onto the scene, and this platform has really just pushed genome engineering into the forefront and made it very mainstream. So if I wanted to be super hipster about it, I would be like, I knew CRISPR-Cas9 before, it was cool. But it's naturally a bacterial immune system machinery where it targets invading viral DNA and then chews it up. It recognizes a 20 or technically a 23 base pair sequence. So it's a very rigid sequence length, but still long enough to denote specificity in a genome about the size of the human genome. It's RNA guided, so that's really the key point of what makes it so different. And because it's RNA guided, it's very easy to reprogram. The CRISPR-Cas9 system actually refers to this complex of Cas9, the Cas9 protein, which is an endonuclease, and the guide RNA, or this short guide RNA, the single guide RNA, that can just be switched out in a matter of like days, as opposed to weeks where you would need to grow up a whole new protein if you were using a zinc finger or a tail base. But really, whichever tool you pick, you can now make double-stranded breaks in a very specific location in virtually any genome. Genomes such as viruses, human cell lines, model organisms like Drosophila, C. elegans, zebrafish, mouse, Arabidopsis, and more economically important species like tilapia, which is a big deal in China apparently, the potato, which is delicious, and even species that are responsible for some of the worst infectious human diseases in the world, like Plasmodium, which brings us to my next chapter actually, which is how we designed and used artificial transcription factors for malaria research. And this was really, I should say that this was done in collaboration with the Lockhart Lab, who are really the malaria experts on campus. We're really lucky to have people like them working here, because without them this project wouldn't have been possible. So if you're like me, you didn't know much about malaria, a little bit quick background. Malaria is a vector-borne disease caused by parasitic protists in the genus Plasmodium. It's, in 2013, there were over 198 million cases of malaria and over 600,000 deaths. The disease is particularly prevalent in sub-Saharan Africa and Southeast Asia. And while there is a cure for malaria, there are only two major classes of drugs available for treating it. And unfortunately, the emergence of drug-resistant parasite populations really threatens our ability to treat the disease. So there's this urgent need to develop new drugs and find new ways of controlling this parasite. All Plasmodium species have a rather complex life cycle that alternate between a mosquito and a vertebrate host, like us. I'll just really focus on what happens in the human body. But it all begins when you're bitten by a female mosquito containing parasite sporozoites in their spit. And actually the first place that it goes into the liver, where it grows up in the liver and eventually escapes the hepatocyte and enters what is known as the erythrocytic cycle, where these escaped merozoites then invade red blood cells, and then thus begins the cycle of parasite growth and cell rupture. And during this time, the patient often experiences fever, chills, and sweating in a cyclical manner. During this time, in the erythrocytes, the parasite can also take an alternate path, which is terminal, called gametocytogenesis, where it differentiates into either a male or a female gamete. And this is important because this is what's actually taken up by another mosquito when it takes its next blood meal, thus continuing the cycle. There are a number of species that infect humans, but Plasmodium falciparum is perhaps the most infamous, because it's responsible for the most lethal form of malaria, with a fatality rate as high as 20%, particularly in young children. This is due in part to this PFEMP1, or erythrocyte membrane protein 1, proteins that are displayed on the outside of infected red blood cells. So our goal for this project was to use artificial transcription factors, or ATFs, as I'll refer to them, to modify the expression of undruggable gene targets in Plasmodium falciparum, because we believe that this technology that we have now gives us an unprecedented flexibility to go after targets that traditional small molecule drugs could not go after. In particular, the target that we wanted to go for was this monoallelically expressed VAR gene that encodes for PFEMP1. So in every Plasmodium genome, it possesses multiple members of this VAR gene, just different variants of it, but only one of them is ever expressed. And the one that's expressed ends up expressing the EMP1 protein, which is then displayed on the outside of infected red blood cells. The reason why this is important is because it becomes an immune evasion mechanic, mechanism that the parasite can use. So typically, our body makes antibodies against these cell surface proteins, but because the parasite is capable of switching it on a regular basis, suddenly our immune response can no longer respond to it, or develop a lasting immunity to the parasite. Furthermore, these infected red blood cells stick to the microvasculature. EMP1 expression also causes red blood cells to be a little sticky, and this becomes a real problem when it starts clogging up your blood vessels, especially in critical organs like the cerebellum. And this is really what causes so many deaths from falciparum infection, is because your microvasculature gets blocked up, and then the organ, which leads to organ failure. The tools that we wanted to use were based on tails with an artificial transcription factor. So we begin with our tail base, which is responsible for the targeting and the DNA binding. And then we can attach, say, either a CRAB, which is a repressive domain, or a VP64, which is VP64 tetramer, which is a activating domain. The project workflow, well, when I look at the slide now, it looks really simple. But you first create your artificial transcription factors, and you transfect them into red blood cells. And then we want to assay two things. We want to see if our proteins actually got into where they needed to go, so we assayed that by immunofluorescence microscopy. And then we also wanted to test gene expression to see if we're actually changing the levels of target gene expression. And we did this by qRT-PCR. Just quickly, the look of the protein. It is this big protein with a bunch of bells and whistles on it, but they're all necessary. So there's the maltose binding protein for purification. There's a cell-penetrating peptide, which I'll get into a little bit later. A nuclear localization signal, so it knows that it needs to get into the cellular nucleus. There's an HA tag, there's an N-cherry tag that we had hoped to use for immunofluorescence. There's the tail, and then infected domain, whether it's a CRAB or a VP64, and then followed by another 6-hit tag. So I really want to focus, so right now I want to talk about the cell-penetrating peptide, which its purpose is really to deliver the protein into the parasite. And the logic behind this is that we were hoping that cell-penetrating peptides, or CPPs, would be a gentler method compared to electroporation, which is typically used in malaria research, for protein delivery that may be extensible to multiplex experiments, and more importantly, to potential therapies, because you really can't electroporate a patient. I don't care how many release forms you have, you can't do that. So to see whether or not it actually got in, we did immunofluorescence, and so this is the control. You can see that the green signal would be what our anti-HA antibody found. You can see that there's some noise level, but you can also clearly see that there really is no green associated with the DAPI-stained parasite nuclei. However, when we added our proteins, I don't know if that shows up very clearly, but you can see that now there's this co-localization. There's this co-localization of green on the periphery of the parasite nucleus. So this gives us a signal that our proteins are actually going where we need to go. As for whether or not they're actually doing something, well, we designed our ATS to go after a single VAR gene and target the upstream sequence, or the UPS. And then so I chose 4 sites that had varying distances from the transcriptional start site here, just to see which position actually had the biggest effect on the gene expression. So our hypothesis was that if you have this red blood cell that expresses PFMP1, if you were to downregulate with a CRAB, you would basically deplete the expression of EMP1 or just knock it out altogether. Or if you have upregulation, the hope is that maybe we can force it to express multiple versions of EMP1, going against and bypass that monoallelic expression mechanism that it typically uses. What we ended up finding was that we got downregulation of the target VAR gene, regardless of the effector domain. So here's the CRAB with a pretty deep fold change. The VP64 also showed a decrease in expression, which was a little unexpected because we had hypothesized that VP64 was an activating domain, so we actually had expected this to go up. Another thing to note is that the target that worked the best, I didn't show the other targets because they really didn't do much better than control, but this target was the closest to the transcriptional start site. It was literally like just 2 bases away from that transcriptional start site. So it could be that if you're going to target this particular gene, you really need to make sure that the target's close to where the transcriptional machinery would bind, or would have to go through. So in summary, we were able to successfully deliver our tail ATS into the parasites in infected red blood cells using our cell-penetrating peptides. We found that targeting the upstream sequence nearest to the transcriptional start site of a specific VAR gene results in downregulation, regardless of what effector domain we used. However, we did find that So this suggests that maybe the ATF binding alone may be sufficient to prevent gene transcription. Maybe we didn't need that effector domain there in the first place. So some potential applications for where this research could take us. If I had another 5 years here, I would probably go after new genes. So there's this new AP2 gene family that has made a big splash, where it's essential for plasmodium development in invertebrates. Specifically, it seems to be the master regulator of gametocytogenesis. So if you can prevent the parasite from forming the gametes, you can prevent transmission into the next mosquito. So it really halts it to just that host. Alternatively, there's also new species. Plasmodium vivax, while not as deadly as falciparum, is much more widespread than falciparum, where about, I think, like 80% of the cases of malaria are attributed to Plasmodium vivax. However, it's also responsible for chronic malaria because it has this other pathway that allows it to just hang out in the liver and escape the immune system completely, only to crop up again like 10 or 15 years later. However, the biggest thing is that it cannot be cultured. So this has really slowed down the amount of research that people can do on vivax. And it's a problem that we would like to address if we had the time and resources to investigate it. So that kind of wraps up the malaria project. And then the next chapter would be Mermaid, which is a de novo multiple motif discovery tool that we came up with that relies on k-mer counting. So at the start of it, Mermaid is a software tool designed to be used in conjunction with Bind and Seek, which is an in vitro binding assay developed by the Korf and Segal labs here at UC Davis. It uses an oligo library of randomized 21 base pair sequences and allows the DNA binding proteins, or DBPs, to bind a full range of sequences rather than just being limited to what's available in a cell. The oligo design is pretty straightforward. You have a 3 base pair barcode that you can use for multiplexing sequencing reactions. There's the random 21 base pair sequence I was talking about, where the protein is supposed to interact with. And then you have a quality control tag that we use to verify the quality of the read itself. So essentially what you're doing is that you're exposing your proteins to all the possible reads it could ever want. And basically you just let them pick out their favorites, and then you sequence it. And so during this time, you're really curious about what sequences it's actually pulling out. One of the things is that Bind and Seek can produce a lot of raw data, but the software solutions at the time, like me, were too slow or computationally intensive to use for a lot of in like high throughput experiments. So like, for example, Meme was one of the things that you would send your data in, and about like a week later, you're just kind of sitting there, you're like, oh, I hope it knows that, I wonder where it is. And so that kind of thing was a bit of a worry, and we wanted to accelerate that. And the way we did that was we came up with this program called Mermaid, which is short for K-mermaid motifs, if you're wondering where it came from. And it's a k-mer counting based method for motif finding. It also has, it doesn't occasionally have a sailor to the desk with your science on. That's not part of, maybe in the next version we'll add that. Essentially what the workflow is that it starts with data acquisition, where you start with Bind and Seek with your proteins of interest. You pull down, you sequence the reads that the proteins bind to, you compare it to a background of unbound oligos. And then you go into the QA and sorting, where you make sure that the reads are good quality, and then you sort them based on the barcode into separate reactions. And then for each one of these barcodes, you then take it into motif analysis, which is a big long pipeline here, where we count the k-mers, and then we score and select them. Then we build the core motifs, expand them, and then we give an output to the user. So the way Mermaid really built k-mers was through the power of friendship, or kind of nearest neighbor matching. So if you imagine that this NIMER, this cat-cat-gat, is the most popular NIMER in the pool. Like, everyone wants to hang out with it. So if you imagine it as this handsome fellow here, what Mermaid would do was find friends that are highly similar to it, that maybe only vary by one characteristic. And then so that they would be a friend. However, k-mers that were a little bit too different would end up not being so they would just be kicked out from the site and not contribute to that final motif. So the friends work together, or then combined into one like super friend, or a motif that has characteristics of all the contributing friends depending on how popular they are. So if we're going back to letters, essentially if we have the most popular motif up here that say we saw it 500 times, and then its friends that vary by one letter in some position, and those occur at a pretty high frequency, but not as high as the most popular one. you end up with what we call the core motif, where the height of the letters indicate how conserved those bases are in a given position. Then Mermaid takes it a step further, where it expands the motifs based on the original data. So actually, once we get the core motif, we go back into the original data, and we try to figure out what goes on the outsides here around that core motif to see if we get some context clues. And so that's the raw data. It ends up looking kind of short ways to either end, and then we get a little bit more insight on perhaps the nucleotide composition that these motifs are normally found, because that could actually be an extension of the core motif. So to see if this actually worked, if our algorithm was sound, we tested the ZIF268. So the ZIF268 is a zinc finger with a very, very robust 9 base pair motif. I'm not going to read it, you guys can read it. But essentially what we were able to pull out is that Mermaid was able to recover that 9 base pair motif pretty well, and with very high enrichments. I know you guys don't look at Mermaid data, so just trust me that this is very high. These numbers are big. But it was also able to find, to the flanking that core motif, some interesting things, like the fact that ZIF268 seems to prefer to bind between 2 T's. And that hadn't been previously reported in any studies regarding ZIF268. So that's exciting to know that it's a little bit more than just, so say if you're looking for a target site, you could say that it's more likely to bind this 9 base pair target site if it was close to a T or preceded by a T. Another protein that we looked at in collaboration with the Knopfler lab was MIS1. So MIS1 stands for the MYC-interacting zinc finger protein. It's an endogenous transcription factor with multiple binding partners, including the oncogenic MYC. But little is known about the actual function as a transcriptional regulator, including its binding motif. So MIS1 has 3 domains. It has this pause domain, which is required for dimerization of the transcription factor. It has a zinc finger domain, which is responsible for DNA binding. And it has this mirror domain, which is the MYC-interacting region. In this talk, we'll really focus on the pause and the zinc finger domains. And our question was, how do the pause and zinc finger domains contribute to DNA binding? It was previously believed that pause doesn't play a role in DNA binding, but most certainly the zinc finger does. But at the same time, people believe that pause was critical for DNA binding, not to actually change the sequence, but for dimerization. So you needed to dimerize in order to actually bind the DNA. That was the initial belief. So they first tested, Knopfler lab first tested the full-length MIS, which revealed this one motif, this first motif. I can tell you that the 2 bottom motifs look completely different, but they're actually reverse complements of each other. So we basically pull these together and say that they're the same motifs. And the enrichment scores are pretty good. However, then they took out the pause domain and wanted to see, does the zinc finger recover the same motif, or does it recover a motif at all? And sure enough, it actually does. And with comparable enrichment scores, except this motif, at least the first 2 look very different from the first full-length motif. So it came to the conclusion that it reveals 2 distinct MIS1 binding motifs depending on which domains are available, or present actually. So the full-length MIS binding motif is this temer here, whereas if you just chopped off that pause domain and were left with the zinc finger, you got primarily this 6 base pair motif instead, which suggests that MIS1 dimerization doesn't appear absolutely necessary for DNA binding, but the presence of the pause domain does affect the DNA binding specificity. So it doesn't seem like the zinc finger is solely responsible for the binding target. And now for something not so completely different, which is, this entire time I hope I haven't given you the impression that the technologies we use are perfectly specific, because they're not. You could give a DNA binding protein a very specific sequence, and because you're a good geneticist, you made sure that there's only one, and it's very unique. And even then, the protein might kind of want to know a little bit more if your protein was anthropomorphic. We all talk to our proteins, right? Late nights in lab, it happens. You try to be a little bit more specific, but whatever, it just goes off and does its own thing, and you wait for it to bind, and it comes back with more than what you expected. So this happens with every platform. Every platform you have, you might have said that it only targets one thing, but it ends up going somewhere else. And these off-target effects are problematic because they potentially lead to lower efficiency of the the protein and increase toxicity. So these off-target effects could end up cutting in critical genes that end up killing your cells rather than just modifying them. In all these, so I talked mainly about, I said that all these platforms have off-target behaviors, and these are for 3 of the popular ones. So zinc fingers, it was found that the longer your arrays get, you get lower on-target activity. And the longer arrays have changing specificity, which makes it really hard to figure out where those off-targets are actually happening. And then in tails, we have the N-terminal polarity, which I'll talk about in a second. And then CRISPR-Cas9 initially had been thought to be very specific, but it turns out that it actually tolerates mismatches anywhere in its target site. And also, unlike the other ones, it actually tolerates indels as well. So this tail polarity, which Josh McAlery, who's sitting back there, he was instrumental in doing pretty much all the wet lab work, but he made this observation where mismatches at the 5' end, or the part of the motif that was associated with the N-terminal end of the tail, were more disruptive to tail binding than mismatches at the 3' end. So it got us to thinking, like, hey, maybe there's something positionally important about how this protein was binding. Maybe there are regions of it where specificity is more critical than other parts. And when we ran our binding seq mermaid pipeline, we found that we could only find enrichment of the 5' motifs. So any motifs that could have been associated with the 3' end were happening so infrequently that we could never recover it, or it was just not specific enough that mermaid didn't think it was a motif in the first place. So basically, if we were to draw a specificity graph on tail polarity, on tail specificity, we would see that it basically goes down from the N-terminal end, where it's highly specific, it's really near perfect, and then the further you get away from that N-terminal end, the specificity goes down. So essentially, if you're working with tails, make sure the most critical part of your sequence is at the 5' end, and then as you go on, you can't count on it to recognize the 3' end. So in conclusion, mermaid is a faster way to resolve binding motifs de novo, and can reveal binding context through motif expansion. We can see a little bit more than what the core motif reveals initially. And we also discovered 2 MIS1 binding sites, whereas we had only been expecting to find 1, and this was really dependent on the presence of the pause domain. And then thirdly, we established the N-terminal, or the 5' binding polarity of the tail binding specificity, where there's this kind of this polarized specificity gradient that is where the N-terminal end is the most specific end, and as you get to the C-terminus, it starts not caring about what it's binding. So all this science really couldn't have been done without collaborations and just the bedrock of having a good lab setting. There were 3 things I wanted to do before I graduated. I wanted to become a better scientist, I wanted to become a better programmer, and I wanted to learn to ride a bike. So the first thing, obviously I like to think I became a better scientist, in no small part thanks to my colleagues and my mentors. I became a better programmer, which I can credit mostly to Ian. I mean, he already claimed it. So that's that. Still working on the bike thing. I'll get there. But yeah, so from the day-to-day, the Korf lab, or the Bioinformatic gurus, if I ever had a question on how to program something or debug or to find that missing semicolon, these are the people I could turn to for help. And even beyond that, just for advice regarding a career path and alternative interests in science. And so there's Ian right there, if you couldn't recognize him, just hidden in the back. And also the Segal lab, who really were the molecular were the basis for all my molecular biological work. I'm really fortunate to be in a group of extremely talented, enthusiastic individuals, because I knew that if I had a problem, I could at least 1, gripe to them about it, and 2, find a solution to it. So just to point out where Dave is, he's right there, and he's also right here. I mean, no one's been able to explain to me how this happens. You know.
There's 2 Daves.
So one Dave's in Australia, and I assume the other one is also in Australia. But also there are a lot of collaborators and funding sources that have also helped a lot. The Lockhart Lab obviously was, with their generosity, we were able to do as much as we did with the Malaria Project. The Knopfler Lab for all their work on this one. The Baldwin Lab, who were a huge help in assembling all these tables. Like they're really experts on that kind of protein biochemistry and purification. Obviously, the University of California for being my home for the past 9 years since undergrad. The biotechnology programs, the designated emphasis in biotechnology. I'm going to shout out a plug here. If you're not in it now, you should join it, especially if you have if you have any aspirations of joining, going into industry, because they're a very supportive group that will give you the insight and the advice that you need and the opportunities that you need to be successful in science beyond academia. Funding from the Bill and Melinda Gates Foundation for the Malaria Project. They were very generous with a pilot grant for this kind of like pie in the sky idea we had of curing malaria. And the National Institutes of Health for funding my fellowship for 2 years. It was a huge help for pursuing my research interests. Obviously, all my colleagues at the university, or UC Davis Integrative Genetics and Genomics graduate group, the UC Davis Genome Center, where I kind of hung out in my cube of solitude or in the lab, and obviously to friends and family who may not have understood what I talked about at all, but understand that it took a lot of hard work and it took a lot of collaboration to get to where we are now. So with that, I'd be happy to take any questions, comments. If you don't have questions, you can always eat too, and maybe questions will come to you.
Did you do all of those drawings yourself?
Yes, except for the Wikipedia logo and the 3D things. Also the photos, I did not draw those people. Alright, cool, thanks guys.
Thank you.
Abby, maybe you can tell about your new adventure that you're going to be doing at Segal Pharmaceuticals. What are you going to be doing there?
So today is my second to the last day here at UC Davis, and starting on Monday, I will be starting an associate scientist position at Sutro Biopharma. I had previously done a postdoctoral internship with them last summer, and had such a great time, and apparently they liked me well enough to take me back. So I'll be doing a lot of cell biology there, which is a different path than what I've done here, but it's still exciting. The group is great. And I think really the most valuable thing that came out of this PhD is just learning how to learn, how to be able to ask the right questions and be able to find those answers, regardless of what specific topics we were looking at. So, and you know, that wouldn't have been possible without my education here. Great. Thanks, guys.